be starting our topic on 1.2, which is the ultrastructure of cells. And so just to dial it back towards the beginning of the semester, remember that there are three domains of life. We have archaea, which are our ancient guys that live in hostile conditions, like our methanogens. And then there's also bacteria. So this is the stuff that can infect you. It's kind of what we think of more common in bacteria. And then there's finally eukarya. So I want to introduce you to maybe some new words you've sort of heard before, but not really. So archaea and bacteria are both something called prokaryotes, and the pro means before, and we're going to say that kary means nucleus. So prokaryotes are the kind of organisms that existed before there was a nucleus. And so over time, evolution occurred, and eukaryotes came along, and eu just means with. So eukaryotes are with a nucleus. So these categories, prokaryotes and eukaryotes, are determined mainly by differences in their structure. So I'm going to just tell you up front that prokaryotes are the simpler of the two. They are primarily single-celled organisms, and what really sets them apart from eukaryotes is that they do not have compartments of any kind. All of their stuff is it's like a studio apartment. There's no walls separating any of the different rooms. So there's no nucleus or any other membrane organelles. Also, these guys are really small, only between 1 to 3 microns large. And an example would be either the bacteria or archaea bacteria. Both are prokaryotes. And they have, again, going back to your meiosis unit, remember that prokaryotes have that single circular DNA, but it's contained in this special area called the nucleoid. And I'm going to add some new things to this category, that they have something called a 70S ribosome, only this kind, and that they have the structure called a pili. So go ahead and get started on your Venn diagram, filling this part out. So these are the characteristics that are unique to prokaryotes only, but it does share some things in common with the eukaryotes. So both cell types have cytoplasm. Okay, they all have some stuff inside. And both eukaryotes and prokaryotes have these 70S ribosomes, although the eukaryotes have an additional different kind. And both prokaryotes and eukaryotes both contain DNA stored in chromosomes, but remember the prokaryotes have that single circular DNA, where the eukaryotes have the multiple linear. And both prokaryotes and eukaryotes at least have a plasma membrane. So these are the traits that each of these have in common. You'll see these questions a lot where you're asked to compare and contrast. So when you're comparing them, these are the traits that they have in common, and these are the things that make prokaryotes unique. All right. So just to review, what is in common? Cytoplasm, ribosomes, although eukaryotes have an additional different kind. Both have DNA that is stored in the form of chromosomes, although prokaryotes only have that single circular versus eukaryotes who have the multiple linear. And both have plasma membranes, whether you're prokaryote or eukaryote at least. But again, prokaryotes are only ever single-celled. If it says bacteria, it's a prokaryote. If it says archaea, it's a bacteria. Again, prokaryotes do not have any compartments of any kind. They're just studio apartments. All the functions occur in one room. They're very small compared to eukaryotes. And again, that they have a circular DNA that's located in something called a nucleoid. And they only have 70S ribosomes, and they have this other structure called a pili. So let's look at this whole thing about ribosomes. So again, prokaryotes and eukaryotes both contain ribosomes, whose job it is to synthesize proteins. So you see this is like your mRNA code, which comes from your genetic DNA code sequence, and it's building proteins. So this is two parts of the ribosome, a large and small subunit. And again, just remember that both of them can contain the 70S version of the ribosome. So let's look at the structure of prokaryotes. So this is an electron micrograph of bacteria. This is really what they look like on the outside, very tiny. So you're going to see this diagram a lot, and you'll be expected to be able to label this whole thing. So first things first, all organisms, if you're a cell, you have some kind of a plasma membrane, and that plasma membrane is what's controlling what's coming in and out of the cell. But in bacteria and prokaryotes in general, they have to have this cell wall on the outside because they're living in the environment on their own and they need that cell wall for protection and support. So when you see these multiple layers, know that the plasma membrane is the one that's kind of on the inside and the cell wall is the one that's on the outside. Again, all cells have some cytoplasm, which we know from our last unit is where the metabolic reactions always take place. And again, prokaryotes only have the 70S ribosome, which is the site of protein synthesis. They make proteins and they are free in the cytosol. You might remember these sort of structures from your Chlamydia demonis. These are the flagella. And again, these are used for movement. E. coli has them too, so this is the specific cell it is. And then these little hair-like things on the E. coli are called pili. So these aren't cilia because this is not a paramecia, this is an E. coli cell. And these pili are used for attachment. So this E. coli wants to probably infect things, so it's going to use these guys to attach either to organisms or something that it wants to digest, or they can also be used in sexual reproduction, but that's much rarer. And then this little circular chromosome in here that's much smaller, this is a plasmid. So remember the plasmids from the biotech unit. These are the things that can carry extra genes for the bacteria. They can also be the carriers for that antibiotic resistance, and you'll see them, there's one here and one here. So these are contain DNA, and they're just much smaller than the larger chromosome. So this would be the single circular DNA that's in the center. Remember that prokaryotic chromosomes do not contain any histone proteins, so they are considered quote-unquote naked. And the area in the cell where they are located is called the nucleoid. So even though it's not separated, they're just calling the area that this is in the cell is the nucleoid. And again, it's not associated with protein, so you might hear it referred to as being naked DNA. All right, so as I said, bacteria and other cellular structures that you're going to see inside eukaryotic cells are really, really small. So the light microscopes that we have in the room can only have a maximum resolution of 0.2 micrometers. So if you do that conversion, remember micrometers is -6 and nanometers is -9. If I take the number that's different between them, 9 minus 6 is 3, so I go 1, 2, 3. That gets me to 200 nanometers. Many prokaryotes and some, not the cells, not the eukaryotic cells, the organism, let me just change that right now. Not eukaryotic cells, eukaryotic, go back. Eukaryotic organelles. So eukaryotic organelles are less than 200 nanometers. So the light microscope will not be able to see them because they're too small. So electron microscopes, which you use to make these electron micrographs, they have a maximum resolution of up to 1 nanometer. So you can see these organelles in eukaryotes as well as the prokaryotes clearly. So all the pictures that you're going to be seeing from this unit are from electron microscopes or electron micrographs. Okay, so let's talk about how prokaryotes reproduce. So for the most part, they don't do sexual reproduction. They mostly reproduce sexually through a process called binary fission. And they're basically making clones of themselves. They're just two cells and they make a copy, and then you have two identical daughter cells. So there's a number of steps that you take to do this. So let's say I'm a little bacteria cell and I have my single circular chromosome in here that is not associated with protein. It's just here in the center of the cell as naked DNA in the nucleoid. So the first thing that's going to happen is that DNA is going to coil. It's going to condense on itself. And then the DNA is going to replicate because eventually it's going to make two daughter cells and all cells need that DNA. So it's going to make itself a copy. And then, just like the chromosomes are pulled in opposite directions, like in meiosis, the same thing is going to happen. They're pulled to opposite sides. And then the cell wall is going to grow because it's going to make a bigger cell that it wants to divide in half. At that point, you get the formation of a new cell wall, and then voila, you have two new identical E. coli cells. So that's the steps in binary fission. Okay, so moving on to eukaryotes, and remember that eukaryotes, in contrast, they're mostly multicellular. That's not actually true, but Ivy says so, so we're going to pretend that that's true. They are the only ones that are multicellular, I can say that. Prokaryotes are mostly single cell, but only a eukaryote can be multicellular. And in contrast to the prokaryotes, where that cell is open, it's like a studio apartment where there's no doors or dividers or walls between any of the different rooms. Now the eukaryotes do have those compartments. They have a nucleus and a bunch of membrane-bound organelles that we're going to talk a lot about. These also tend to be much bigger, where the prokaryotes were between 1 and 3 microns in size. Eukaryotes are at least 10 times that size, between 10 and 100 microns. And basically, an example is literally anything that's not a bacteria or archaea is going to be a eukarya. So that includes the kingdoms of Protista, animals, and plants. We are all eukaryotic. And instead of, again, because these eukaryotes have compartments and the prokaryotes don't, our DNA is in a nucleus instead of being in a nucleoid. And in addition to the 70S ribosomes, eukaryotes also have the 80S ribosomes. They're just a little bit bigger. Okay, so those are the ones that are unique characteristics of eukaryotic cells. So why did cells evolve in this way? Like, what's the benefit of doing this at all? So one benefit is that instead of being sort of free-floating and around a cell, enzymes and their substrates, and these are the guys that do the chemical reactions, are going to be very concentrated. They can be because you're stuffing them into a room, right? So that's a benefit. And the other thing is that sometimes the substances that you want to use in a cell can be very damaging. And so having compartments, having these organelles gives you a means to contain those damaging substances. So like an example is the lysosome, which breaks down macromolecules. It digests things. And it can digest the cell if those enzymes were out in the cell itself. So they get contained inside the lysosome where they can do their job without hurting anything else. So think of it like, I don't know, what's damaging in your house? Like you could have like your kitchen, right? You don't want the kitchen stuff with all the sharp knives and the hot stove in the baby's room, right? Because that stuff could really hurt one of your little brothers or sisters. But if we contain it in the kitchen and the baby's not allowed in the kitchen, then everything's fine. The other thing is that within organelles, special conditions can be maintained. So an example is like lower pH. So in the lysosome, those digestive enzymes, and this is just a means to probably protect the cell, they only work at lower pHs. So the lysosome is specialized so that it can do that. So you can do that when you have separate rooms. And then the fourth reason is that, organelles with their contents can be easily moved within the cell. Like right now, you carried a bunch of stuff to school in your backpack. How much harder would it be to move all of your papers and your books and your pencils if you had to carry them individually one by one, right? It's easier to just toss everything into the bag and go. So same thing with the cell. If I need, this function to be on the other side of the cell, I just take the organelle with everything in it and move it. So those are the benefits of having the compartments. So let's talk about what all of these are. So we'll start with animal cells. So just like a prokaryote, we have a plasma membrane which controls what exits and enters into the cell. Same as in all living things. And same as in all living things, you have the cytosol, which is just sort of the space in the middle, and that is where all your metabolic reactions are going to occur. And remember that eukaryotes have these 80S ribosomes, but they have a specialized function. So these guys generate proteins for proteins that are going to stay inside the cytosol. They're going to stay inside the cell. So the 80S ribosomes do protein synthesis for proteins that are going to stay inside the cell. They're not going to go anywhere, so those are specialized. And again, eukaryotes have a nucleus, and this is sort of going to be the big giveaway when you look at the pictures of what you're looking at, prokaryote versus eukaryote. And inside that nucleus, you have the DNA that is packaged into chromosomes, which sometimes you'll be able to see on those micrographs. And those chromosomes are wrapped around histone proteins. And remember, eukaryotes can have multiple, and they're linear. They're no longer circular. Okay, and that is always in the nucleus. So in eukaryotic cells, you have these things called mitochondria. And they have like sort of this, they look a lot like bacteria, but they have this sort of like crisscrossy stuff in the middle called cristae, and we'll talk more about that in a minute. And the mitochondria is what's generating energy for you. So mitochondria is the site of respiration, and it is the organelle that generates your ATP energy. You could not survive without it. And because these are related to bacteria, they also contain the 70S ribosomes. So eukaryotes have both 70S ribosomes, which only do protein synthesis inside the mitochondria, and then they have the ADS ribosomes, which do, and when they're free like this, in the cytosol, those proteins are going to stay in the cytosol. Okay, so mitochondria does respiration, generates ATP energy, and also contains those 70S ribosomes. So you might see these other structures around, and maybe they're lysosomes. And the way that you can tell these apart is that they stain really darkly. And then the lysosome is special because it is the only organelle in the entire cell that is not a double membrane. So the lysosome is only a single membrane. So your plasma membrane that you have around, your cell is a double membrane. The mitochondria also has a double membrane, and your nucleus and everything else in here is going to have a double membrane. Only the lysosome has a single membrane, so it's special that way. And its job is to basically digest macromolecules. It digests kind of everything, proteins and lipids. If it gets in there, its lower pH is going to chew everything up. So that's the job of the lysosome. And so then we have this should say rough. Let's change that to rough. No, not that one. So here we have the rough endoplasmic reticulum. And I know it's the rough endoplasmic reticulum because it has these ADS ribosomes attached to it. That's what these little black dots are here. So these ADS ribosomes are synthesizing protein that's going to stay inside the cell. But these guys that are attached to the endoplasmic reticulum are making proteins that are destined to be secreted, that are going to be kicked outside the cell. So they're either going to go into the membrane or kicked outside the cell. So that's the job of the ER. And you'll always be able to tell the ER because it is right against the nucleus because it gets mRNAs directly from there, and it's going to have these little dots all over them. So that's how you'll be able to identify that. What's usually further away and off to the side, but looks really similar to the ER, is the Golgi apparatus. So proteins travel in vesicles from the rough endoplasmic reticulum over to the Golgi, where that Golgi makes vesicles, which I guess are represented here, and then they go over to the membrane, where they either get embedded in the membrane or they are secreted outside the cell. So the last thing, and I'm sorry, this is really tiny, animal cells have something called centrioles, and so these help form microtubules during cell division. They're really hard to see on electron micrographs, and I don't think you'll see it often. They usually sort of point them out and tell you, but sometimes they'll have this star and sort of line shape here. And again, you only find centrioles in animal cells. Okay, so study this micrograph for a minute and tell me what you think these different structures might be. Okay, so now that that's done, so these are the mitochondria, and you can tell that they're the mitochondria because they have this sort of the cristae in between them. The nucleus is usually the first thing that people can identify, and here they're telling you that this is a centriole. Okay, and so we always know the easy one is a cell membrane. For you call this plasma membrane, which is always on the outside. So this one was a little tricky because it has these folds, which makes it look a lot like the ER, but this is the Golgi because it's not right against the cell, or the nucleus, I'm sorry. So the ER, the rough ER, when you see it, it's going to be right up against the cell, and hopefully they don't try to trick you like that. And then this one would have either been a lysosome or a vesicle. And it's weird that it's so far away from the Golgi. But okay. Okay. So you might see some questions about people asking you to distinguish between a mitochondria, which is on this side. And I don't know why it's not labeled. Hold on. Okay, we'll just leave it. Hey. Okay, a mitochondria on this side and a prokaryote on this side. So what is in common between both of them? They look really similar, but the mitochondria has these structures called cristae. And you can usually pick out a mitochondria because there's obviously other cell structures in the area. This one looks like a rough ER because, again, you see these folds with the little tiny black dots. What is in common to both of them is that they both have the 70S ribosomes. They each have a single circular chromosome, although I think in the mitochondria it's curled up somewhere in here. And both of them have that double membrane, the double plasma membrane. But the prokaryotes are a little different because they have, in addition to the plasma membrane, they have this cell wall that's on the outside, and you can see it's like more darkly staining. And in addition to that, they have these extra structures like the flagella and the pili that you never see in the mitochondria because the mitochondria doesn't need to like move around or do anything. Okay, so that's how you would tell the difference between them. All right, so let's move on to plant cell structures. So plants are eukaryotes, so they have membrane-bound compartments just like animal cells do. They also have a plasma membrane that controls what exits and enters. They have a cytosol where the metabolic reactions are taking place. They too have a mitochondria, again, that you can pick out because of those cristae in the center. They also have the lysosome, which again is the only organelle to have a single membrane that digests like waste and macromolecules. And then you have, again, the nucleus, which with its DNA packaged into chromosomes with histone protein. They too have a rough endoplasmic reticulum, and they also have those ADS ribosomes, which generate the proteins that stay inside the cell, versus the endoplasmic reticulum with its rough ribosomes that generate proteins destined for either the membrane or secretion. So once the proteins are made by the rough ER, they get passed to the Golgi apparatus, which then makes those vesicles to transport the proteins either again to the membrane or out of the cell completely. So all of that is still the same for plants, but plants need water, right? So they have a large central vacuole, which helps them to store the water. And then plants are much more rigid than animal cells, so they also have a cell wall around them to help give them some protection and structure. And then the other thing that the cell wall does is it helps prevent excess water intake. So they don't want to like overwhelm the cell with water. If it's out there, it sort of blocks too much water from coming in. It helps to maintain that homeostasis of water. And then, of course, you know that plants have chloroplasts, which have these like weird sort of streaky shapes in them. It's how you can tell them apart. And the chloroplasts are the sites of photosynthesis. That is what makes glucose that they can then give to the mitochondria to do respiration and generate that ATP. So, you know, plants are pretty self-sufficient. It's pretty cool. All right, so go ahead and look at these guys and label the different structures that you